De-extinction, reviving extinct species with advanced genetic techniques, is moving from science fiction to reality, as scientists use CRISPR, cloning, and ancient DNA to bring lost creatures back. The woolly mammoth, a colossal creature that once roamed the vast icy plains of the northern hemisphere, has long captured the imagination of scientists and the public alike. Towering above most modern animals, these giants were perfectly adapted to the harsh, frigid conditions of the last ice age, with their shaggy coats, curved tusks, and immense size. For thousands of years, they were a dominant presence in the Siberian tundra, shaping the landscape and supporting entire ecosystems. But then, in a dramatic turn of natural history, the woolly mammoth vanished from these frozen expanses. Their disappearance, which occurred around 4,000 years ago, marked the end of an era. The reasons for their extinction are still debated. Some point to climate change and the warming of the planet, while others highlight the impact of human hunting. Regardless, the tundra has remained empty of these giants ever since, their absence leaving a void in the ecosystem. Now, in a remarkable blend of science fiction and reality, researchers at Colossal Biosciences and Harvard University are working to bring the mammoth back, at least in a new form. By extracting DNA from remarkably well-preserved mammoth remains found in the permafrost, and using advanced gene editing tools like CRISPR, they are attempting to insert key mammoth genes into the genome of the Asian elephant, the mammoth's closest living relative. This process involves painstakingly identifying the genetic traits responsible for cold adaptation, such as thick fur, layers of insulating fat, and specialized blood proteins. The ultimate goal isn't to create a perfect clone of the extinct mammoth, but rather to engineer an elephant that possesses many of the mammoth's most important traits. Imagine an elephant with a dense woolly coat, a robust build, and the ability to thrive in sub-zero temperatures. These mammoth-elephant hybrids could one day roam the tundra, equipped to survive and even flourish in the cold, open grasslands of Siberia. But why bring back a version of the mammoth at all? Scientists believe that reintroducing these hybrids could have profound environmental benefits. By grazing and trampling the land, these animals could help restore the Siberian grasslands, which in turn would slow the thawing of permafrost, a process that releases vast amounts of greenhouse gases into the atmosphere. In this way, mammoth-like elephants could become unlikely allies in the fight against climate change, helping to preserve the Arctic's delicate balance. The journey to resurrect the mammoth is filled with scientific, ethical, and ecological challenges. Yet the potential rewards, restoring lost ecosystems, combating climate change, and pushing the boundaries of genetic science, make this one of the boldest frontiers in modern biology. As researchers continue their work, the world watches, wondering if the tundra will once again echo with the footsteps of giants. Passenger pigeons once filled North American skies but vanished due to human activity. Revive and Restore is working to bring them back by editing the DNA of band-tailed pigeons, their closest relatives. By pinpointing and inserting key genes, scientists hope to recreate the passenger pigeon's unique traits. Success could restore lost ecological roles like seed dispersal and rebalance forest ecosystems. The return of these flocks could transform North American woodlands once again. The thylacine, also known as the Tasmanian tiger, once roamed the dense forests and rugged mountains of Tasmania. With its distinctive stripes and dog-like appearance, it was a symbol of the island's wild heart. Tragically, relentless hunting, habitat loss, and disease drove the thylacine to extinction in the early 20th century. The last known individual died in captivity in 1936, and since then, the thylacine has become a haunting reminder of how human actions can erase entire species from the planet. Yet, stories of mysterious sightings and the enduring hope of its return have kept the legend of the Tasmanian tiger alive in the public imagination. Now, a team of scientists at the University of Melbourne, in partnership with Colossal Biosciences, is working to bring this lost predator back to life. They are meticulously sequencing the thylacine's genome, piecing together its genetic code from preserved specimens. To achieve this, they're using the Dunnart, a small, mouse-like marsupial, and one of the thylacine's closest living relatives, as a genetic template. This approach could allow them to recreate the thylacine's unique traits, from its striped coat to its powerful jaws. By carefully editing the DNA of the Dunnart using advanced gene editing tools like CRISPR, Researchers hope to insert thylacine genes and eventually produce animals that closely resemble the extinct Tasmanian tiger. This process is incredibly complex, involving not just the transfer of genes but also the challenge of ensuring that these genes are expressed in the right way to recreate the thylacine's appearance, behavior, and ecological role. If successful, this would mark one of the most ambitious de-extinction projects ever attempted. The project is filled with scientific and ethical challenges, but its potential impact is enormous. Restoring the thylacine could help rebalance Tasmania's ecosystem, where the loss of this apex predator has had lasting effects on native wildlife and vegetation. It could also serve as a powerful symbol for conservation, showing that science can not only prevent extinctions, but perhaps even reverse them. If the thylacine is revived, it would be a groundbreaking milestone for both science and conservation, a testament to human ingenuity and our responsibility to heal the wounds of the past. The return of the Tasmanian tiger could inspire new hope for endangered species everywhere, reminding us that extinction doesn't always have to be the end of the story. The dodo, a flightless bird from Mauritius, became a symbol of extinction after disappearing in the 17th century. Scientists have sequenced its genome and are exploring pigeons as surrogate parents for dodo embryos. By editing pigeon DNA, they hope to hatch dodo-like chicks and revive this iconic species. The dodo's return would be a powerful symbol of restoration and scientific progress. It could inspire new conservation efforts worldwide. The Pyreneian Ibex, a mountain goat from Spain and France, was declared extinct in 2000. In 2003, scientists briefly revived it through cloning, though the clone survived only minutes. This breakthrough proved cloning's potential for de-extinction, despite setbacks. Researchers are refining techniques to improve embryo health and survival rates. The Pyreneian Ibex's story offers hope that extinction might not be forever. The great auk, a flightless seabird, vanished from the North Atlantic in the 19th century. Scientists are using razorbills, their closest relatives, as potential surrogates for genetically engineered great auk embryos. By editing razorbill DNA, they hope to hatch chicks resembling the extinct auk. Restoring the great auk could help rebalance marine ecosystems and right past wrongs. This project highlights science's power to address human-driven extinctions. The quagga, a uniquely striped zebra from South Africa, was hunted to extinction in the 19th century. The Quaga project uses selective breeding of plain zebras to revive quagga-like traits. By choosing zebras with reduced striping, they're producing animals that closely resemble the extinct quagga. While not genetically identical, these zebras restore lost appearances and ecological roles. The project shows how selective breeding can help recover lost biodiversity. The Heathhen, once common in eastern North America, vanished in 1932. Scientists are using genetic engineering to introduce Heathhen genes into greater prairie chickens. The goal is to recreate the Heathhen's traits and reintroduce it to its former habitat. Its return could boost plant diversity and ecosystem health. The project demonstrates genetic engineering's promise for conservation. The moa, towering flightless birds native to New Zealand, once roamed these lush forests in great numbers. For centuries, they were the undisputed giants of the land, shaping the ecosystem with their presence. But after the arrival of Polynesian settlers, the moa vanished, driven to extinction by hunting and habitat loss. Their disappearance left a void in New Zealand's natural world, altering the balance of plants and animals that had evolved alongside them. Today, scientists are on a quest to bring these giants back. In high-tech laboratories, researchers have painstakingly extracted ancient DNA from moa bones, some buried for centuries beneath the forest floor. By comparing this genetic material to that of the emu, one of the moa's closest living relatives, they're piecing together the moa's genetic code, unlocking secrets that have been hidden for thousands of years. Using cutting-edge gene editing tools, scientists are now attempting to insert moa genes into emu cells. The hope is to create living moa embryos, which could then be placed into surrogate emu mothers. If successful, this would mark the first step toward reviving a species that has been extinct for centuries, a feat that would push the boundaries of science and conservation. The challenges, however, are immense. The process is fraught with technical hurdles, ethical questions, and ecological concerns. Yet the potential rewards are equally great. Restoring the moa could help rebalance New Zealand's unique ecosystems, allowing native plants and animals to thrive as they once did when these giants walked the land. If scientists succeed, it would be a groundbreaking achievement, a true marvel of modern science. The return of the MOA would not only rewrite the story of extinction, but also inspire new hope for conservation efforts worldwide. Imagine a future where these ancient giants once again roam the wild landscapes of New Zealand, reshaping the environment and captivating the world with their remarkable comeback. Steller's sea cow, a massive kelp-grazing mammal, was hunted to extinction within decades of its discovery. Scientists are analyzing preserved DNA and considering dugongs or manatees as surrogates for edited embryos. By editing their genomes, they hope to birth animals resembling the extinct sea cow. Its return could help restore kelp forests and ocean health. Reviving Steller's sea cow would be a milestone for marine conservation. De-extinction raises tough ethical and ecological questions. Should we focus on reviving lost species or protecting those at risk now? Critics worry about unintended consequences like invasive species or new diseases. Others argue de-extinction could distract from urgent conservation needs. Supporters believe it can restore ecosystems and repair human-driven damage. As science advances, we must weigh risks and rewards with care. The future of de-extinction and conservation depends on wise, responsible choices.